Okay, today's lesson is about meiosis. It is topic 3.3. Here are our syllabus points. Please make sure you have a read. Same deal as always. All right, let's rewind to start with. We have previously talked about mitosis. Mitosis is topic 1.6. We talked about it in terms of cell replication, remembering that mitosis is the replication of a nucleus to produce two genetically identical daughter cells. Okay, we aren't talking about mitosis this time. We are talking about meiosis. Now, meiosis is a process where one diploid nuclei is turned into four haploid nuclei. Remembering that diploid, we have two of each of the chromosomes, so two chromosome 1, two chromosome 2, etc. And in haploid, we only have one of them. Those haploid cells are gametes, okay? Gametes being the sperm and the egg or the sex cells. And we know that the halving of the chromosome number is really important, allow for fertilization to create a diploid cell when they are fertilized. So the chromosomes of the parents are going to be different to the chromosomes of the offspring. Now, this is going to allow for variation because it's going to actually add different genes in, as opposed to, say, mitosis or binary fission in unicellular organisms, where it's just an identical photocopy of the genes of the parent. So not really much variation there. Now, mitosis occurs in every cell in the body. However, meiosis only occurs in the reproductive organs, so in the testes and the ovaries. Mitosis is for growth and repair, but meiosis is solely to produce gametes, those sex cells, no other reason. Mitosis has one diploid parent cell producing two identical diploid daughter cells, but meiosis has one diploid parent cell producing four haploid daughter cells. So we need to know the difference. It's really important. Right, meiosis is incredibly complex. It is a multi-step process. It's like mitosis, but there's an extra division. And there's uncertainties as to how it developed, but it's important to know that for the origin of eukaryotes, it allowed the fusion of gametes and fertilization in sexual reproduction, which can only really be done by complex organisms. Okay, remembering that a diploid cell has two of every single chromosome. So in this particular case, we've got an XY right there. So it is a male. And if we have haploid gametes being produced, the sperm and the egg there, say that person was back here was a male, then we are only producing sperm and they only have one of each of the chromosomes there. And this particular male is contributing an X to this sperm, so therefore it could go and join together with an egg and then make a female offspring. Okay, prior to meiosis, we have to remember that DNA can replicate through the interphase, so the S phase. It's going to start as a 2N or a diploid cell. It goes through meiosis 1, so the very first stage of this multi-step process, and it will straight away split into two haploid cells. Then during meiosis 2, there is another replication, and that haploid cell sort of replicates itself, and it's like a mitotic replication. And remember, those final cells, those daughter cells, are actually the gametes that we are talking about. The stages of meiosis are very similar to the stages of mitosis, right? Same letters that we can remember, same sections: prophase, metaphase, anaphase, and telophase. All right, remembering that at interphase, this is our normal cell functioning. We've got a 2N cell, so we've got this red one here pairs up with this yellow one, and this red one and this yellow one. So in that case, it'd be paternal and maternal of each of those. At that S phase, the DNA synthesis occurs, so the sister chromatids are now present, so they're kind of those back-to-back little Y or X shape, In that prophase 1, chromosomes condense and they coil really tightly. During the first metaphase, this is where the difference lies. It's not like mitosis. In metaphase, here, these guys match up with their partner and line up at the equator. In mitosis, they just line up at the equator. They don't have to find their partner. But in meiosis, they do find their matching pair. Now, in anaphase, the spindle fibers attach to the entire chromosome and pull them apart from their pair. We aren't splitting sister chromatids. We are simply pulling them apart from their matching partner. So in that telophase, we've got the spindle fibers shortening. We're pulling the whole chromosome to the opposite pole, and this is known as disjunction. So two haploid daughter cells are formed. They're haploid now, okay? So the sister chromatids are still intact, but only one from each of that homologous pair are in each cell. So there is cell division, but the chromatids have not split yet. Now we head into that meiosis 2, and that's kind of called the mitotic like division because it does look like mitosis. Now the same thing's happening, it's just only with one possible part of the pair in there. So we've got them lining up again at that metaphase, they line up at the equator. At anaphase, the spindle fibers attach, they shorten, they pull them apart, two separate cells in that final telophase. So at the end, we've got those 4 haploid daughter cells, and these are our gametes, so the sperms or the eggs. So if we go back to that very first metaphase where the pairs are lining up at the equator, we've got these paired chromosomes and they're known as bivalents. And in the early stages of metaphase 1, they're kind of lined up next to one another. And what they do is they kind of hold hands and play footsies, okay? And they can actually cross over parts of their chromatid. Now, because the gene sequence is linear, they can swap genetic material, and that's kind of a mutual exchange of genes. And this shuffling of alleles leads to increased variation, and crossing over of both maternal and paternal features means that we can kind of almost pass on our grandparents' traits to the offspring there. All right, now there is a possibility that something goes wrong, and in non-disjunction, that's what's happening. So this is our normal thing that should be happening. We've got one of each pair going into the to the first sets of haploid cells, and then we have one of everything in our final daughter cells. However, sometimes homologous chromosomes fail to separate, so the partners don't want to be torn apart. And in that second meiosis phase, sometimes these sister chromatids don't get pulled apart. And what it can lead to is known as trisomy, where we can potentially end up in an offspring with 3 of a chromosome, and monosomy, which is where in an offspring we can only end up with 1 of a chromosome. And this is only possible if you get 1 X. You cannot survive with only 1 chromosome 1 or 1 chromosome 20, but you can survive with 1 X. That's it. Now, if we have a look at this one, this is the partners, the homologous pairs didn't want to separate, and we're going to end up with gametes with nothing useful in them for that chromosome, and that's not going to be a viable pregnancy. And in this situation, we have the right thing happening at that first meiotic division, but by here, we've actually got a situation where the sister chromatids have not split and have ended up in the same gamete there. So one reason that this can happen is advanced maternal age. The eggs have been waiting a long time in the ovaries for maturation and release, and therefore can lead to a risk of non-disjunction. Things just don't work properly. And this, we'll just pause this and clear some of these. This is a karyotype from a patient who has trisomy 21. There are 3 chromosome 21s there, and that is classically known as Down syndrome. So trisomy 21. Now, we can find those types of abnormalities by looking at the fetal cells before that fetus is actually born. So between the ages of 16 to 20 weeks, we can collect an amniocentesis. And then what they do is they use an ultrasound in order to guide a large needle into the amniotic sac and collect the amniotic fluid from around the fetus. It contains fetal cells, so we can do genetic testing to make sure that all the chromosomes are in the right place. Same thing can happen with parts of the placenta. You can go in at about 12 weeks, and it's called a chorionic villus sample, or a CVS, and you can go in, take a small chunk of the placenta, not enough to harm the baby, and you can sample the fetal side of the placenta as opposed to the maternal side of the placenta. There is a tiny risk of pregnancy loss for both of these because they are invasive, meaning they do actually enter into the female body. Right, here are our syllabus points. Please make sure you have a read.